to follow up a little bit from what Graham was saying about the China Scholarship Council Innovation Platform. That it has a real possibility to be able to support interactions between our institutions. So I sincerely hope that it is successful. And if it is successful, we will be able to support interactions between the different groups that are here today. I'll leave it there and then start by asking Caroline Dye to present on liquid biopsies for the treatment of lung cancer. Thank you, Caroline.
Okay, it says screen sharing has stopped as the shared window is closed. I'm not quite sure what that means. So,
oh, that's a new one on me.
I don't think you can see. Can you see my screen now?
Yes, we can.
Okay. The trouble is I can't see my slides for some reason. Let me just. Are you seeing my slides?
Yes, we can see your slides now.
And are they on full screen?
They are. Yep.
Okay. All right, then I'll get started. So it's a wonderful pleasure to be here with colleagues from China, and I'm really looking forward to the opportunity to collaborate with the Shanghai scientists. Today, we're talking about liquid biopsies. I'm going to concentrate on the treatment of lung cancer. And just in the next 15 minutes, tell you two research stories. The first, how we use circulating tumor cells to predict risk of recurrence in non-small cell lung cancer after surgery. And the second, a newer story, to talk about methylated circulating tumor DNA and how we may be able to predict the newly defined subtypes of small cell lung cancer. Are my slides moving?
Yep.
Yep, okay. So you probably know that up to half of patients who have surgery for non-small cell lung cancer can relapse. And really what we wanted to know is, can we predict the risk of this recurrence after surgery at the point of surgery using a liquid biopsy? And could this inform the frequency of minimal residual disease monitoring with circulating tumor DNA? So the story started back in 2016, working with Philip Crosby here at the University of Manchester. And we wanted to ask, are circulating tumor cells actually quite an early event in non-small cell lung cancer, traditionally thought of as a late event, but actually, could they be an early event? And what we did was not look for circulating tumor cells in a peripheral blood sample, because we already knew that using cell search, which only finds epithelial circulating tumor cells, you rarely find them in early stages of non-small cell lung cancer if you take the blood from the arm. But instead, our surgeons took blood samples just before they removed the resected tumor. So they took them from the pulmonary draining vein in the cancerous lung. And here we found that there were circulating tumor cells even as early as in stage 1. And if you count those circulating tumor cells in the pulmonary vein, you can find just in this first pilot study that we did, that first circulating tumor cells are out and about early. And also they may be able to predict the risk of recurrence of disease at surgery. So we then went on to work with Charlie Swanton from University College London in our Lung Cancer Centre of Excellence from Cancer Research UK in the TracerX consortium, a consortium across multiple sites in the UK. And again, we took the pulmonary vein blood just as the patients were having their tumours removed from the lung at surgery. And what Charlie's team was able to do was to do sequencing of the resected tissue, but also sequencing of the metastatic relapses. And our job was to look at these circulating tumor cells all the way through the progression from surgery, follow-up, relapse, further follow-up, progression and completion. So the multi-region sequencing was done in Tumor in London, and the circulating tumor analysis was done here in Manchester. And whilst there were several aims of this consortium to define lung cancer evolution, to determine impact of clonal heterogeneity, our job was to predict the risk of recurrence. And so we took our blood, the pulmonary vein blood, we enumerated those CTCs with cell search, but we also isolated those single CTCs. And we asked, are those pulmonary vein CTCs genetically linked to relapse, as well as can the number of those CTCs predict the recurrence? So we did this in 100 prospectively recruited patients where the PV-CTC protocol was available. And what we saw, confirming our pilot, that the 48 of the 100 patients had at least one cell search CTC in their pulmonary vein blood. And when you counted those CTCs, it confirmed that the number of those circulating tumor cells at surgery would predict the risk of recurrence. So I want to take you through a case study next. This is a patient case study. A patient arrived with an invasive adenocarcinoma in the lower right lobe, and then they had their surgery. And Charlie looked at 3 regions, multi-region sequencing of 3 regions of the resected tumor. We isolated 28 pulmonary vein CTCs, and then the metastasis that arrived later was also sequenced. So if I run through this study, here mutations are present, absent, or we didn't have reads. And if we look in the regions 1, 2, and 3 of the resected tumor, you can see these clonal mutations. You can also see some mutations in
Caroline,
each of
do you want to hold on a second? Oh, it's muted now. It's all right.
Okay, and then I'll just repeat this one. So in the 3 regions of the tumor, different regions of the tumor, there were clonal and subclonal mutations. In 3 of the isolated CTCs, we also see these matching clonal mutations, matching subclonal mutations, but you also see here private mutations that were not found in the tumor, but were found in the CTCs. And then when we sequence the metastasis that arose 10 months later, you can see that these metastases also contain these private CTC mutations. And in fact, 91% of the mutations were detected in the metastasis. The slides seem to be moving by themselves. I'm not quite sure why. But anyway, you can see that the CTCs are part of the metastatic clone in this phylogenetic tree. And what was interesting is that there were 14 mutations shared between these CTCs and the subsequent metastasis, including an inactivating driver mutation in a tumor suppressor gene. And this is a gene that acts to inhibit tumor migration. And we also know that the lower expression of this gene is linked to poor prognosis in this disease. So we really feel that if we look at these CTC mutations at surgery, we may be able to inform on the panel of mutations used using circulating tumor DNA to follow those patients up so that we can intervene earlier as their tumor reemerges with recurrence. So the second story I want to tell you today is about small cell lung cancer. This is an aggressive neuroendocrine disease where 80% of patients present already with metastatic disease. Many of them have a good response to their platinum-based chemotherapy, but they relapse very quickly, almost universally with acquired chemotherapy resistance. Biomarker assessments in clinical trials in small cell are still very rare, and that's mainly because serial tumor biopsies are very, very challenging. Immunotherapy has been licensed with some responses, but we don't have a validated predictive biomarker, and we need new therapies for small cell very urgently. So the really big change in the field of small cell research came in 2019 when myself and colleagues across the world, led by Charles Rudin at Memorial Sloan Kettering in New York, pulled together data that showed that there were molecular subtypes of small cell lung cancer, not based on their genomes, where P53 and RB1 are hallmarks, but otherwise genomes are very heterogeneous, but by subtyping based on transcription factors. And there are 2 major transcription factors, ASHL1 and NeuroD1, which are neurogenic transcription factors, but also some non-neuroendocrine subtypes based on YAP1 and PAL2F3. And this particular very rare subtype comes from a different cell of origin, a tough cell, rather than the neuroendocrine body cell, which is the typical cell of origin for small cell lung cancer. And this is important because preclinical studies by ourselves and folks at MD Anderson and others showed that these different subtypes may respond differently to different targeted therapeutics. So the ASHL1 subtype to a BCL2 inhibitor, the NeuroD1 subtype perhaps to an aurora kinase inhibitor, the PAL2F3 to a PARP inhibitor, and a new subtype defined later called the inflamed subtype that might be the subtype that's responsive to immunotherapy. So it's really important to think about functional heterogeneity and subtype vulnerabilities as a way of going from what is now homogeneous chemotherapy treatment, perhaps towards personalized medicine for small cell lung cancer patients. So we're wondering how useful circulating tumor DNA can be in small cell lung cancer. And we showed in a patient cohort of nearly 70 patients that we could generate whole genome NGS libraries from each circulating free DNA sample from peripheral blood. We could use an aliquot to look at the copy number changes by shallow whole genome sequencing. And then we used a second aliquot for targeted enrichment of a panel of 110 genes, frequently mutated in small cell. And we've enriched these for DNA damage repair pathway genes because of the trials upcoming and ongoing for DNA damage repair and cell cycle therapies in clinical trial. But I just show you here the copy number for routine patient monitoring in ctDNA. You can see this typical copy number profile for small cell lung cancer at baseline. You can see that it disappears at the end of the first treatment with chemotherapy in a responding patient. It's still gone at follow-up number 1, but at follow-up number 2, that acquired chemo resistance has kicked in, and this copy number profile is back. And you can see it there at clinical relapse. So it would appear that the copy number monitoring may be actually a quicker indication of relapse rather than the clinical imaging and clinical signs. But we wanted to ask, could we get more sensitive in our monitoring of small cell lung cancer? And could we use circulating tumor DNA to identify which molecular subtype a patient was, given that biopsies in this disease are really very difficult, necrotic, small, and difficult for research? So we've started to think about circulating tumor DNA methylation. We know that this is an epigenetic marker. We know that it occurs early in tumor development. These cancer-specific changes in methylation are early. We know that aberrant methylation occurs at much higher incidence than somatic driving mutations, and hence it's not surprising that CFD and methylation profiling is much more sensitive compared to mutation detection. And here in Manchester, this is a busy slide, I'm not going to talk to all of it, but just to say that we've worked on a new workflow for CFD methylation that has some really useful features. It has a circular adapter, it allows barcoding so we can pool samples, so that we can have high throughput of samples. And this was mainly for early detection studies. It has unique molecular identifiers that allow us to correct errors during bioinformatics. It has a T7 RNA polymerase, which also reduces PCR-associated sequencing errors. And the new method that we've developed is suitable for genome-wide and targeted approaches. And we can build additional assays in using this workflow, and notably cfDNA and copy number changes from the same tube of blood. And for our methylation assay, we use a methyl binding protein. And this is a much milder process than the usually used bisulfite method. And it allows us to protect and maintain the fragmentation patterns in circulating free DNA. And that allows us to have a more precise identification of circulating tumor DNA. And this was a workflow led by Dominic Rothwell and Alex Clipson in the lab. So I just want to show you some early data using this new ctDNA methylation. Just to give you some background, methylation is associated with inactivation of tumor suppressors in small cell lung cancer and deregulation of some of those lineage-specific genes like that transcription factor I mentioned, NeuroD1. We know that EZH2 is overexpressed in small cell where it regulates methylation via activation of DNA methyltransferases. And we know that promoter DNA methylation levels are high in small cell lung cancer. We also know that DNA methylation can identify epigenetically distinct small cell subtypes, but we don't know how these map to those transcription factor subtypes that I mentioned earlier. So the early work we did shown in these heat maps is when we looked at some published data on ctDNA methylation in small cell, in normal tissue and in tumors. And then we compared that using our method, this T7 method, in models that we make from circulating tumor cells of patients with small cell lung cancer that we call CDX models, and a few models which we made from biopsies from small cell patients. We can see that this maps pretty well to what's already published in 50 of our preclinical models. But also when we make a circulating tumor cell derived explant model, where we take the blood from patients, we isolate the CTCs, we put the CTCs into immune compromised mice, and we grow a tumor that we call a CDX. And when we compare the methylation profile of those CDX with the same, a parallel blood sample that we generated the CDX from, and we look at the cfDNA, we can see these concordant ctDNA methylation patterns in the CDX tumor and in the circulating free DNA from the same patient, which would suggest that the method is working well. But really exciting, I think, is we can look at this cfDNA and we can show that it has increased sensitivity when we look at the methylation patterns. And this just shows the tumor prediction score based on that ctDNA methylation profile. As we go through the chart, we can go from normal healthy controls through the small cell lung cancer patients. And in particular, we can pick out stage 1 disease of small cell lung cancer. So we can pick up patients who were not detected using that copy number assay that I showed you. So the methylation is more sensitive as we thought it would be. And also those methylation scores from our patient's ctDNA are also prognostic for overall survival. Perhaps the most exciting thing of all, though, is when we look at cfDNA methylation patterns, we can distinguish the different major subtypes of small cell lung cancer. And we've done this in preclinical models, those CDX models made from patient CTCs and grown in mice. And we can predict whether a patient is a NeuroD1 molecular subtype or an ASHL1 subtype. And there's just one patient that is neither, one of these rare subtypes. And we can do this in our preclinical models, but we can also have some preclinical models where we've got the matched cfDNA and the tumor, and we can again predict the NeuroD1 from the ASHL1 subtypes. And this frequency of subtypes is very similar to published data identified in the our identification of methylation in the blood sample in cfDNA with immunohistochemistry data from tumors. So we really think, and we've submitted the manuscript now, that we can use cfDNA methylation not only for more sensitive detection of small cell lung cancer, but also to tell which molecular subtype it is. And this in future may allow stratified medicine approaches in small cell, which after decades of chemotherapy, which works very briefly and then the patient relapses, we think is a real step forward. So the take-home messages from this part is that our new workflow for ctDNA methylation is a robust method. We can see patterns in small cell lung cancer patients' blood. The new assay increases sensitivity, and it allows us to define the molecular subtypes from a blood draw. The last slide I want to show you is I think this idea of liquid biopsies in lung cancer is so appealing. We know biopsies in lung cancer are challenging, not without risk for the patient. CTCs and ctDNA are prognostic, predictive, can be used as pharmacodynamic biomarkers and to monitor a minimal residual disease, and as I've shown you in small cell for molecular subtyping, but they're also being used now for early detection. And I just want to mention some amazing work that Phil Crosby has done in Manchester, detecting lung cancer early using low-dose CT scanning based in the community in some of our socially deprived populations around Manchester. So folks get a letter, they come to their supermarket car park, they have a low-dose CT scan, and we also collect their blood for research. And we're using a new technology called HDSCA to look at circulating tumor cells. We take the blood sample, we put all of the cells that are nucleated on a sticky slide, and then we use immunofluorescence assays to identify the very rare cells. And I just want to show you one really exciting piece of data where we've used this new CTT assay to look at the number of CTCs in the blood for patients who've come through this low-dose CT screen. We had 108 cancers coming through. 35 of the patients had false positive low-dose scans. 52 had indeterminate scans, and 41 had negative scans. And you can see here, these are the number of the CTCs measured on these slides. And you can see that when they were blinded for outcome, they have a higher number of CTCs, 108 CTCs here, for the patients who had a low-dose CT scan and an early tumor, a stage 1 tumor, was diagnosed. And that's bigger number of CTCs than the controls who didn't have cancer and the controls who had a low-dose CT scan that turned out to be a false positive. And the work that we're doing with thousands of blood samples collected through this low-dose CT scan screen is we're combining this new CTC assay with ctDNA mutations and that methylation assay that I just showed you. And of course, the hope is this time in non-small cell lung cancer that we can pick out with very good accuracy early detected cancers that can have surgery with a really high chance of cure. So I hope I've shown you some examples of how we use liquid biopsies in lung cancer, and I'm very much looking forward to hearing the next talks, and if there are any questions, I'd be happy to answer them. And just thank you to my team, the Cancer Biomarker Centre group. We're about 100 folks. I want to acknowledge Fiona Blackhall, a talented, brilliant thoracic oncologist with which I do all my small cell lung cancer research. And this is the new building that Rob Bristow mentioned in the introductory section that's going up. And this is myself standing on the 10th floor of the building, really looking forward to moving back here and continuing with our translational research. Thank you.
Thank you very much, Caroline. It's a while since I've heard you give all of the talk out at one go, so it's impressive. It's impressive what you've been doing. I was going to ask if people had questions. If not, I did have one or two rustled up my sleeve. So actually, when you were talking about the in the first part of the talk about lung cancer, and you noted that the circulating tumor cells had mutations which were not present in the primary tumor, but were present in the metastases. I noticed that the profile of those mutations in the circulating tumor cells was different. So some samples had certain mutations and other samples had other mutations. Yes. Does that allow you to stratify? Does that allow you to? So
it's a wonderful question, Keith. What we need to do now, we're just waiting to see whether we've got a new grant from Cancer Research UK to do a lot more on actually looking at more and more single CTCs to see how heterogeneous they are and what that actually tells us. So at the moment, just the number of them predicts pretty well which patients are going to recur. But if we look at those private mutations, you know, if we do enough of this, are the private mutations that really do indicate what type of tumor is going to recur and what to treat it with. So I think there's lots of future potential of the circulating tumor cell profiling to really address the heterogeneity issue and to actually ask the question, is there some information in amongst that heterogeneity that really can inform treatment? And the difference between the CTC risk of recurrence is we can do that at surgery, whereas the ctDNA monitoring, it picks it up early, but not as early as surgery. And I think, you know, what we want to do is take the benefits of CTCs at surgery and then the ease of monitoring with cfDNA. And maybe some of these CTC mutations should go in the MRD panel that we use for the circulating tumor DNA monitoring test. So I think the combination of the two, everyone often asks, you know, why would you do CTCs when you can do ctDNA? Because it's easier, and it is, but it's not quite the same as having the whole cell. And it doesn't really get you to the heterogeneity problem. So a lot more work to do. It's quite expensive at the moment. And I'm sure that in Shanghai, folks are interested in liquid biopsies, and we're about to hear. But I think there's lots of opportunities here in the early detection space, in the MRD monitoring space, and in the small cell lung cancer clinical trials, because small cell is actually quite common in China, more common than it is in the UK. So I think there's lots and lots of potential here for collaboration, which would be wonderful.
Perfect. I was going to also ask you a question about Notch signaling and small cell.
Oh my goodness.
Lung cancer, but maybe I should ask you. Maybe I should just make the trek down the road and ask you in person.
Well, I can make some high-level comments, Keith. So we're really interested in Notch signaling. What Notch signaling does in small cell lung cancer is it makes the neuroendocrine cells, which are the majority, transition to non-neuroendocrine cells. And those non-neuroendocrine cells have a very different biology. And I think to target small cell, you actually have to have a therapeutic strategy that kills both the neuroendocrine and the non-neuroendocrine cells. And just to give you one example of what the non-neuroendocrine cells do, Notch-driven, is they can pretend to be endothelial cells and make their own blood vessels. It's called vasculogenic mimicry. So Notch signaling in small cell is really important.
Okay. That touches, then I should have a conversation with you.
You definitely should.
Because we were doing some work on stem cells and mesenchymal stem cells and driving them endothelial using Notch.
So there you go. Same biology. Yeah.
We should have a conversation. All right. It's always good when you find out what your local neighbour's doing.
There we go. Pandemic doesn't help. Yeah.
All right. Perfect. So I think we'll leave it there for Caroline, and then it's a real pleasure to ask Ziming Li to talk on to give his talk on clinical applications of liquid biopsy in lung cancer. So Professor Li.
Okay, thank you. So the honorable chairman and the dear professors, my name is Ziming Li. I come from Shanghai Chest Hospital. So it's my honor to be here to share my presentations. Okay, so I will share my slide. So today my topic is about the clinical applications of circulating tumor cells, the CTC, as liquid biopsy in lung cancer. So as we know that the molecular biology of the lung cancer is evolving with the pressure of the treatment. So the intratumor heterogeneity represents a major problem to effective cancer treatment and the personalized medicine. So the re-biopsy is very important. However, in some cases, the patients refuse the re-biopsy. So the liquid biopsy is a good way to capture the molecular heterogeneity of cancers. So in the liquid biopsy, we have the CTC, we have the ctDNA, exome, and the microRNA. So, but how do we choose? So that's a question. So as both the CTC and the ctDNA technologies evolve, they have similar as well as distinct clinical applications. They also have their relative biological and technological strengths and weakness. Actually, I think the CTC just a computer cell, so we can do more research on the CTC. Yeah, so several studies have shown that the CTC counts provide the prognostic information, including the integrating prognosis, assisted detection of reaction to anti-cancer drugs, and the biomarkers of the drugs. So we can combine some staging factors to obtain more accurate outcomes to determine if the patient needs the adjuvant chemotherapy, and we can test the cancer recurrence. So the papers published in New England and the JCO, we have seen some results. So we can see a paper published in JCO in 2011. We can see that the number of the CTC was higher in patients with stage 4 non-small cell lung cancer compared with the stage 3A and 3B. And we can see that the patient with fewer than the 5 CTCs compared with more than 5 CTCs, the patient had better the PFS and the OS. So consider the first and second time points together, and we can see that 61 patients with fewer than 5 CTCs at both the time points before and after the chemotherapy, and 9 patients with 5 CTCs at one of the time points. We can see that the both time fewer than 5 CTCs have better PFS and OS compared with the patients with more than 5 CTC. Furthermore, I think the molecular of CTC are also clinical relevant. Some paper published, we can see that the single CTC-based sequence can be used to study the genomic profile, and we can study the evolutionary history of a small cell lung cancer. So we can get more information for the clinical doctors. Other studies show that the molecular events involved in colon cancer progression, and we can provide the potential CTC biomarkers that can help. We can develop new therapies to tumor metastasis and tumor repair. So here comes 2 questions: how to determine if a cell in the blood is a really tumor cell, and how to achieve reliable single cell sequencing for the CTCs. We have the technologies shown in this table. I think the most popular CTC isolation technology has evolved antibody-mediated capture of cancer cells. We can see that it's the cell search technology. This technology makes use of the attack of antibodies against the common epithelial cell surface markers like the EpCAM. And we also can use the high throughput, the microscopic scanning of the blood specimen to find to screen the CTCs. And the other technology about the isolation technology are based on the physical properties of cancer cells. The most popular in the cell search technology, the cell search uses the protein marker combinations. We can use the EpCAM positive, CD positive, CD45 negative markers, but it's not cancer specific, and it can perform the follow-up like the RNA detections. However, some benign cells have EpCAM positive cells, so it's not a perfect way to detect the CTC. So how about the CT research on lung cancer? It seems that we don't have so much studies as much as the breast cancer and the colon cancer. So we should use a new technology to detect CTC. As we know that the energy deregulation is a hallmark in cancer cells. The Warburg effect is a common feature of cancer. The PET scanning images use reagent-labeled glucose to detect malignant tissues with high glucose intake in the body. So we can propose the idea that we can directly detect the high metabolic active cells in the body through samples and then verify if they are really tumor cells through the single cell sequencing. So we published a paper in PNAS in 2017. The topic is about the detection and the sequencing of real tumor cells in the perfusion and the blood based on the glucose intake. So in this paper, we use the 2-MBDG, a glucose analogous, as a marker to indicate suspected cancer cell. So after the sample is filtered, the red blood cells are corrected and the cells are added to the chip. And then we use the combined 2-MBDG, the CD45 and the ETD1 antibody to mark the cells. So the suspected tumor cells were removed for single cell sequencing to verify that they are tumor cells. So in this slide, we can see that we will pick up the CD45 negative and the 2-MBDG high suspected cells. That's the suspected tumor cells. This is a picture about the high-throughput screening image system. We can see that the CD45 negative and the 2-MBDG high cells in this picture. So this picture that is we detect the suspected tumor cells, and then we picked up and to do the single cell sequencing. So in our research, we can find the we just picked up patients about the lung cancer. This is a lung cancer patient, the adenocarcinoma 34. This is a treatment-naive patient, and this patient had EGFR 19 deletion mutations. So we just in the pre-infusion we find the 8 cells, 6 with EGFR mutations. So we can see that these 6 cells are really the cancer cells. And then we also find just a patient, 2 patients, EGFR mutant and the KRAS mutant. So we just find the high 2-MBG intake cells in the blood. And we also confirm with the sequence that they have the same mutation with the primary tumors. So it's about the patient 1. We can find that a totally 5 high metabolic active suspected tumor cells we detect in the pre-infusions. The 3 cells with the KRAS G 12C mutations, but the other 2 cells are the wild type. So in order to clarify whether the remaining 2 of the 5 suspected cells are tumor cells, we did WS in all the 5 cells. So we can see that we can see there are also genes related to the mutations after clustering with the other cell levels. So we can see that, we can confirm that these 2 cells are also tumor cells. And then we use this method, we can see that the detection sensitivity is superior to the cytology diagnosis. In 3 patients, we don't have the confirmed the cytology result, but in the prefusion, we can find that the high metabolic cells have the mutations. So we can confirm that these 3 patients have the metastasis. So we also find that this patient 6, we don't find the clinical drug resistance at the time of the sample collection, but we can see that we have the T97 mutation in this high metabolic cells. So after a period of time developed, the patient has the drug resistant reactions. So in addition, we also know that the more metabolic phenotypes markers have been identified. Several studies have shown that the tumor cells can also perform the oxidative phosphorylations for cell energy. So we propose the idea that more metabolic markers can identify tumor cells that take other energy metabolic pathways. On the other hand, it improves the sensitivity to detect tumor cells. So we can understand that the relationship between energy metabolism and the prognosis of tumor cells. We published a paper in the 2019 in the Nature Communications. The topic is about the accurate prediction of the topic is about the liquid biopsy-based single-cell metabolic phenotyping of lung cancer patients for informative diagnostic. So this slide shows that we combined with the 2 metabolic markers. The first one is the 2-MBDG, and the other one is the resin. It's a redox indicator. And we use combined biomarkers to detect suspected cancer cells. So we have the we have the 2-MBDG high, and then we have the resin high cells, and then we have the third one is the both high cells. So in this group 1, we can find the patients with lung cancer. They diagnosed with pathology in primary lesions and prefusion. But we also find that some patients, the mutation gene consistent with the primary lesions. But the patient 2, the primary lesions was wild type, but we have found that the tumor cells in the patient 2 had EGFR mutations, and the cells driving gene mutation were not detected in either primary lesions or prefusion in 4 cases. So we did the CNV test. We also found in the group 2, we can find the primary lesions were diagnosed as lung cancer, but no clear tumor cells were found in the prefusions. The pathological diagnosis was unclear. And the patient number 5 and number 22 also had 3 high metabolic active subgroup cells. And we also performed the CME test. In the group 3, it's the clinical diagnosis suspected of lung cancer, but no cancer cells were detected in the primary lesions. So the 21 and 23 were all found to have 3 high metabolic active subgroup cells, but no related mutation driving genes were found. So we also conduct the CME test. So we also test the mutation in both CTC and primary tumors in 7 patients. The 7 patients in this study have no gene mutations in primary lesions and the prevention, but cells with CMV were detected. So it's confirmed that's the tumor cells. It's very interesting that we can find the PFS results of the 3 subgroups of patients with EGFR mutant. The high resolution group and the 2-MBG and both the 2-MBG and the resolution high group have a worse PFS, have a better PFS combined with the 2-MBG groups. We also find that the genotypes of the patients with different metabolic phenotypes are similar, but the results of iron sequencing suggest that there was a significant difference between the 2 groups of cells in different signaling pathways, such as EMT pathway signals. So we also published a paper in PNS in 2021. The topic about the HK. We use a new biomarker HK2 deserves a novel CTC population associated with poor prognosis in lung cancer patients. So we use the HK2 to resolve the HK2 high and the CK negative CTC. We can see that the HK2 high and the CK negative CTC is a prevalent population in both the blood samples with CTC positive counts. The single cell shows that the HK2 high and the CK negative CTCs had a smaller size but consistent CMV profiles compared with the CK positive counterparts. And then we also find that the single cell analysis shows that the HK2 high and the CK negative CTCs have a smaller but consistent copy CMV profiles compared with the CK positive counterparts. The single cell transcription profile reveals that the CK expression levels of CTCs are independent of their EMT status. We also find that in the clinical application of the lung adenoma patients with HK2 high and CK negative CTCs, we can see that the PFS of the treatment-naive adenocarcinoma patients with positive CTC counts, we can see that it's segregated by the percentage of the HK2 high and the CK negative CTC in the blood. So how about the futures? We will use the two problem will be used and will be resolved in the future. The first one is the methodological optimizations, and the second one is how to make better use of in the clinic. So this is a summary of the slide. First, this is the first time we find a fast and intuitive high throughput detection method for the GTC and at the single cell levels. And then we also find that this method had high clinical sensitivities. The third one is that we can detection of the driving gene mutations in the clinical values. So this is my presentation. Thank you for your listening.
Thank you very much, Professor Li. I was just wondering if people had questions.
Okay.
Well, first, congratulations on a lovely talk, and I think your talk really exemplified how much more we have to learn about circulating tumor cells, and they can tell us a lot more than we already know. And I think the metabolic profiling in the single CTCs is really impressive. So congratulations on that. I also think validating a liquid biopsy test in the clinic, it's really quite difficult. It needs really robust methodology, it needs really large numbers of samples, and it needs independent site validation. So I think that's food for thought as well, because I think if we could both validate a test in Shanghai and in Manchester and we both get the same answer, then that's a good test. So I think there's some discussions that could be had about how we use each other's abilities in CTCs, because not many labs actually in the world have really good quality expertise in CTCs, and I think we both do. So I think that's a real possibility for the future.
Thank you.
Okay. I was just also curious. You were obviously identifying CTCs, obviously finding mutations. Are you using that to then select the therapies that the patients are receiving? Is it influencing therapy choice?
Actually, I mean, we just as we know that the East Asia patient have more prevalence of the EGFR mutants, so we just select the patients with the EGFR mutant in the primary tumors, and we just test this patient. So it's either way to detect the suspected the tumor cells, and we can do the sequencing to confirm that that is the if it's also had the EGFR mutants, and then we can see that we can conclude that this is the really tumor cell.
Yes, so that was, it was more done to allow you to validate your method that you've actually picked.
Yeah.
When you were faced with that array of cells, you've picked the tumor cell and not something else. Okay. All right.
Okay.
So would the future hold, now that you are definitely picking tumor cells, would you from that do the experiment the other way round, the screen the other way round? Don't select patients with no mutations, but just take any patient and then based on your screening of CTCs, choose your therapy for your patient.
Okay, yeah, that's a very good question. Actually, we just had some data, so we just collect the patient samples, the blood samples. We just do the all the cells to the 10x genomics. We just use the we just test 4 patients. We just send the blood samples to the company, and we just do the RNA sequencing for all the cells, and we find a new way to translate the RNA data into the CMV data. So we just if we can establish this method, I think it will be used widely for the commercial companies. Of course, this is not published. I hope in the future this paper will receive.
Yeah. All right. I think we probably ought to say thank you at this point and move on to Professor Ren ZHAO and ask Professor Ren ZHAO to give his talk on the clinical and scientific researches of colorectal cancer. Professor ZHAO.
Okay, can you see my PPT?
Yes, we do. Yes, no problem.
Okay, thank you. Good morning, Professor and Case Bernard. I'm honored to be here to attend this symposium and the fourth, the Shanghai Jiao University and the Manchus Cancer Research Center and the University of the Manchus. I'll be delighted to take a part in the conference and let me introduce our center, our experience about the colorectal cancer and clinical work and the basic research in our hospital. I'm from the Shanghai, the region hospital. I'm Professor Dr. Ren. Yeah, let me do it. And in our hospitals, and we have before the three the professors, our teacher is etc. And there's especially for Professor Zhu and the Professor Yu. He is a very famous and the professor in China. Yes, is also the chief in our department. And it's so, and our department has around the history is more than the 17. And we took a leading part in the laparoscopic surgery, and also in a new adjuvant radiochemotherapy and preserving the surgery in China. We also own a lot of academic award and appointments. This included in the four area. So that's we introduce the main job. That's including in the three areas. The first is we focus on the early and the diagnosis of the colorectal cancer, the opportunistic the screening for colorectal cancer, and the staging of colorectal cancer and preoperatively. The second is the development and the innovation in the surgical technique. And the technique and including the laparoscopic and the single port laparoscopic surgery and technique, and advanced robotic assisted surgery, and also we use the laparoscopic surgery combined with the intraoperative the radiotherapy. The third area is the scientific research. The major is in the sensitivity of the target therapy for colorectal cancer and the tumor and the molecular market in the research. And we in the clinical work, and we especially in the operation, also the surgical area or the medicine area. And the surgical is the laparoscopic surgery, is robotic and robotic assisted rectomy. The first one. The second is the combined and the radiotherapy, chemotherapy is a new adjuvant and the therapy. And the post-operative, the radiotherapy, and the post-operative in the chemo and the radiotherapy. The second area is the target engines, such as the VEGFR, EGFR, and the PD1, PD1L, the third, and is the minimally invasive, and the process and the functionality is such a. The first way and special in and established the standardized earlier diagnosis the system for colorectal cancer. This is our and the complete and established complete process of the earlier diagnosis of the CRC. We first developed opportunistic the screening of the CRC in Shanghai when we compared with the normal population the screening and there was no statistic and the difference in the positive the rest of the phobic and the earlier diagnosis of the polypus. And however, the difference in the earlier detection of the colorectal cancer is significant. And the second is the way first carry out the process, the preoperative, the staging in our country and improve the oncological outcome. And we have characteristics is in the laparoscopic and the single port laparoscopic surgical and technique, and also include in the robotic assisted surgery and the intraoperative the radiotherapy. And the way development and the major the development is one is from the rectomy to and the laparoscopic the surgery, the first stage, and the two stages is the transition from the convention and the colorectal surgery of the laparoscopic surgery to and the port reduced laparoscopic surgery, and then to and the cells, the single port, and then to the nose, and also the included the 3 stage. And
不好意思打扰一下，赵主任，您那个 PPT 请用全屏播放。
Okay, no problem. Okay, 好，行吗？
哎，好了。
好。And from 2008 until now, and our group finish about 10,000 cases of the colorectal surgery, and including this is including about the 5,000 cases of the laparoscopic surgery, and after the 5 years, 5 years ago, we began and use it and reduce and approach of the laparoscopic surgery, and so we has in the reduction of the invasiveness through the process in the surgery. And we used the 5 years ago, we used the port reduced laparoscopic surgery and the cells. This is a less trauma and minimally invasiveness and transit to cells. And this is sure that the largest number we finished is the largest number of the port reduced laparoscopic and the colorectal surgery, and both at home and at world. And we finished in the cells in the single port laparoscopic surgery is including in the TATME, the trans, the anus TATMEs, and also we used the nurses and then to the nurses. Is in this sure that is the scariest and dearest, the fast track the surgery. So and we about used the 10 years and we exploration of the colorectal surgery. You can see that from the 2009 then to 2020, this including the 3 stage conventional laparoscopic surgery, and 2010 is the 3 port, the reduced port and the laparoscopic, and the 2030s we began use the cells, and the 2020 we began use the noses. So we finished in the 2017. We finished this 1 center. We finished a single center open label in no inferiority the randomized control trials. That's including the 200 cases. 200 cases. They are the baseline the characteristics. There's no including baselines, including the intraoperative and the postoperative outcomes, and the pathology outcomes. There's the results. The results shows that the short-term, the feasible, and the safety, and there's the reduced and the positive post-operative pains, and the further follow-up is needed to confirm is around the results. And 2 years ago, we began the motor center and clinical trials about our cells. And 2000, you can see the photo. We used our cells, the technique, and finished in the venous, the colorectal surgery. And about 1 year, we finished about 10 cases and notes. And also, we used the robotic assisted surgery, the way to finish our surgery. And this is sure that delicate dissection and the protection and the preservation of the pelvic nerves and complete more than 5 cases of the robotic assisted CRC and surgery. You can see that we used the robotic and the technique. This shows that the 3Ds and the high definitions, and you can see and they are very good and automated. So we get in China we get 2017 person of the year of the robotic surgery. And then we also use the robotic surgery and combined with the intraoperative the radiotherapy. It's sure that the integration of the technology and the process of the localization and the list complication. And completed in the first case in Asia Pacific and the second case in the world in the 2012. This is the case report is sure. Now, and now we are used the new technology. We use and we are export intraoperative the technique. This is technically advanced. Advantage is the worst, the smallest, the miniature of the X-rays, the generator, and the combined and the laparoscopic, and also can combine the advanced robotic to achieve the true minimally invasive intraoperative radiotherapy. So, this year we finished nearly in the 6th case, and in the late and advanced, local late advanced and colorectal surgery. It shows that advantage of the express the minimally invasive intraoperative the radiotherapy and cooperative with the laparoscopic and the advanced robotic to achieve and minimally invasive intraoperative radiotherapy. And the application is can and enter through the 1 centimeter the trunk, the word, with the very small word, and the less the breathing and the very quicker in the recovery. And also is the very clear division and accumulate a current and the localization and the convenient operation. So it's very feasible and it's easy to operate and in our surgery. So in the meantime, so we also take a part in the formulation of the colorectal cancer related diagnosis and treatment and the specification of the expert consensus. You can see this, and we the JCO and the 1 paper, and the JHO and the 1 paper. This also is in the combination therapy. And as then, we are introduce our scientific research and work, and it's a major on is the sensitivity of the targets therapy for colorectal cancer and the tumor and the molecular and market research. There also we undertake a full general projects of the National Nature Science Foundation of China, and more than 10 local projects, and accumulate later research funding is more than 10 million yuan in China. You can see this are the projects about our scientific research, the basic research. And this is the paper in the peanuts is the major in the targets, the proteins, the neutralization is the strategy to regulate the tumor sensitivity to the immunotherapy. The second is the paper is in the cancer research in 2019. Is the title is the cell circle dependent facility of the PRPS1, the fuel neurotic the synthesis of the promote tumor genesis. Now, this is our the paper is graph here. This is the third paper, the major third paper. Yes, it's opened in 2021. Yes, it's the fourth the paper one is. And also we the major work in ETF5, the series, the research, says about character about the 5 the paper, is including the 5 ET5 to promote CRC the angiogenesis of the proliferation by the target, the PDGF, the BB, the targeting the tumor the cells driving the CCL2, the could overcome the the resistant ETF5, the positive the feedback loop of the ET5, the beta acting the CCR, the X to promote the radiographic the resistant. Also we are as the editor of the chief, the co-editor of the several academic monography related to colorectal cancers. So I then finished our presentation. Thanks the Professor Kinser.
All right, thank you very much, Professor Zhao. I was going to open it up for questions, but one thing that was crossing my mind whilst I was listening to this is actually I think a good colleague of Caroline and I, Andrew Renahan, might actually be a very good person for you to talk to. So Andrew is also a surgeon, a colorectal surgeon, and I'm conscious that the pair of you might actually have an awful lot more to talk about than other pairs that we have here today. So I'll certainly take that away as something that with Catherine that we should try and orchestrate, something for us to arrange for you to, Professor Zhao, to have a conversation with Andrew Renahan, because I can see obvious overlaps between your work. But
the other Keith, the other thing, sorry to interrupt, the other thing that is quite a predominant research theme in the colorectal surgery. There's quite a few of them now, lab-based scientists and academic surgeons working on peritoneal disease. So there's a large effort in that area as well. And Sarah O'Dwyer, I don't know, Dr. Zhao, if you look on the Christie Hospital website, Sarah O'Dwyer is the lead for surgery rectal surgery, colorectal surgery, and a big effort, as I say, in peritoneal disease. So there is a very active team linked into the translational scientist. A new academic coming up is called Jorge Barrucio, who's very interested in colorectal metastasis to the liver as well. So there's actually quite a vibrant research team there, and I'm sure that would be a really good bunch of folks for you to talk to who are very interested. And Rob Bristow obviously is coordinating that as well, Keith.
Yeah. I'd forgotten that they're interested in the metastasis across the peritoneal cavity, aren't they?
That's
and
right.
the genetic changes that happen as it spreads around the peritoneum, isn't it?
So I really enjoyed your talk. I think you know, the innovations in surgery, sometimes you know the lab-based scientists forget that the surgeons are the ones who cure cancer, and I think, you know, it's really important that we get academic surgery research absolutely on the front page.
Yeah.
Could I just quickly chip in, and probably as part of the follow-up, and Catherine is here, and Rachel and Georgina, and four of us could get up, you know, get together and get Andrew and Sarah, or maybe other relevant colleagues' details together, and to share with Professor Jiao and Catherine and Professor Jiao, if it's okay. And could we have a copy of this presentation so we could actually share it with our colleagues who have not been able to attend this this workshop, but directly relevant to this area of research? Then maybe we could identify a mutually agreeable kind of a time slot and to get them together to talk a bit more in details. What do you think, Keith and Caroline, please?
I think the other important individual there is Malcolm Wilson. So
Malcolm
Wilson, yeah.
Okay.
There are Sarah Dwyer, Malcolm Wilson, Andrew Renahan, Jorge Barrucio. Those are the four, I would say.
Yeah. So the surgery one, Sarah and Malcolm, I'm actually within the Christie, I don't know. So actually I'm glad you were here, Caroline. I would never have picked those up. I'd have picked up Andrew and Jorge, but not the others.
Yeah, it's a very strong team, actually.
Yeah.
Georgina, are you here? Yeah. Good to see Georgina is here. Rachel probably already got off, and
she's there.
Yeah.
So
Okay.
we'll pick that up.
Yeah, we'll pick it up. Yeah.
I think Claire is there as well, and Claire will be able to assist.
Yeah, that'd be great, you know, Georgina, and I'll get in touch and get our colleagues, you know, email address and their full bio, and then we could share it with Catherine and get it forwarded, you know, to Professor Jiao and his team as well. So we could organize a sub workshop, you know, either could be before Christmas.
Yeah,
ideally.
The other thing that we are doing is we are trying very hard in a UK-wide cancer research accelerator grant which is on colorectal cancer to grow CTC organoids from colorectal cancer patients. It's really, really tough. It might just about be doable. We tried to make the circulating tumor cell models from circulating tumor cells from colorectal cancer patients, and we tried really hard and was not possible to do it. So I think, you know, that the liquid biopsy space is mainly ctDNA for colorectal, where Alberto Bardelli's work has shown how useful that can be in charting emergent drug-resistant clones. But yeah, we're trying to make the little organoids from the CTCs, and we have some success, but it is really technically hard.
Caroline, just to jump in there, thank you. Yes, I've picked up, and I'll speak to Georgina and get her to link up with you, Helen, to organise the follow-on. Sorry, I was having some technical difficulties coming off mute and video, so yeah, here. Unfortunately, I do have to drop very shortly, but I really found all the talks this morning really interesting, so thank you.
Yeah, and then the other thing in the colorectal connection is, you know, I know the French group very well that made the colorectal CTC cell lines. But actually, when you look at that, you know, it's from a very small number of patients who had an enormous circulating tumor cell burden, who are very unusual, actually. So the cell lines are there, but they are from a very unusual patient, I would say.
Happy that discussions are going on well. And Professor Zhao, I think maybe later I need to introduce you about the CSC Innovative Platform. It was sponsored by the China Scholarship Council, and actually Shanghai Jiao Tong University School of Medicine has signed the agreement with the University of Manchester. So we will set up the joint PhD education, and as well as the sending the young scholars to University of Manchester for their research training. So we submitted the application to the CSC Beijing office, and we will wait for the result. The result will come out in January, I think. And so if you are interested, it would be happy that you can share with us your PowerPoint, and the Manchester partners, they will also introduce some of the related scientists for the future collaboration. Is that correct, Helen? Yeah.
Yes, absolutely. Yes. We just make sure that we have all the information ready, and get all the professors, kind of have time to review each other's profile, publications, what they have been doing, and hopefully, and we could arrange a dedicated, tailored workshop for them to be able to exchange information and also research ideas together. Yeah.
Okay. Okay, you are welcome. Yes, I hope to have a chance to establish the relationship, also the relationship to exchange our experience and our work, and in colorectal and the surgical, also and the basic researches, and the DNA is the technique is also very good. Probably in our center, we also use this in the technology in test in the late case, in the later case, the followed up, and you use this technology, and we are seeing the results. And maybe in the future we the paper to appear. Yes. I thought it's a very good. It's very good technology. Yes, thank you.
All right. I'm conscious that the Caroline and Ziming, that there's a sort of obvious overlap between you two. And before we finish today, maybe there is a few thoughts of where we could start thinking about for the future. It is just an obvious overlap, I think, with you two.
I mean, my thoughts are, if there is a PhD scheme that is coming on board, we would welcome to have a Shanghai student come over and do a PhD on liquid biopsy and lung cancer. That's one option, I think. And possibly, it could also send maybe a student will want to do some time in China and move over and work there as well. So I think training of young folks in liquid biopsy is one obvious place there we can collaborate. I think the other thing is, maybe we want to set up, I don't know what Professor Lee might think, but, I have a team of maybe 30 people who study CTCs in lung cancer, one way or another, making mouse models in small cell, or the small cell space is a really good one for us to collaborate on, I think, because there are so many CTCs in small cell. We can make animal models, we can do all sorts of things there. And as I say, in China, there are a high number of patients with small cell lung cancer. So I think that area would be great to collaborate on. And in the non-small cell space as well, I mean, there's so many overlaps. We can probably have a whole day or at least a half day where our two groups come together and actually, in more detail than you can possibly show in 15 minutes, go through the liquid biopsy space that we have both in Shanghai and Manchester. And maybe we need to set up a liquid biopsy half day between the two labs, because I think then we could really begin to see where we could be having added value for patients. It's sometimes, I've traveled to China many times. I had my first clinical fellow actually was Chinese. She came on a scholarship to our lab, and she published the first paper from our lab on CTCs. It was Zhang Meihu back in 2013, I think, 2014. If you look at the Cell Search paper on small cell CTCs, this was our China fellow. So there's already a good history of working with China in CTCs, and I would love to do more.
Yeah, that's great.
Perhaps the action is we just have to set up a half-day symposium between the two labs to really look at it properly.
That's exactly what was just going through my mind, Caroline. So if the innovation platform comes through, and I think I'm not putting words into Catherine's mouth here and saying that we're hopeful. I think we're on good ground. So if that comes through, the ability of PhD students to come and visit, for postdocs to come and visit, will be in there. Actually, there's something else, Catherine, which that I'd love Helen and myself and you to talk about. I was talking to others. I've discovered that there is another China Scholarship Council funding scheme, but it works in the opposite direction. So it's a way of getting Caroline to Shanghai rather than Ziming from Shanghai to Manchester. But there is a different funding there is an alternative funding mechanism that will allow, well, maybe not you, Caroline, but your staff and students to head in the opposite direction in a sponsored format as well. So I feel like that there's a bit of homework for myself and Catherine and Ellen to do here in trying to work out that other funding scheme and to actually close this circle. So if we get the innovation platform that brings people from Shanghai to Manchester, have the other one which will take Manchester to Shanghai.
I mean, the other obvious way to collaborate is, we can share the protocols that we have, and have parallel sample collections in Manchester and in Shanghai, and then actually cross-validate those tests. And I think that could work, but I think we probably need the half-day symposium to figure that out.
Yes. Yeah. Exactly. That sounds. Yeah. That's definitely. That sounds like a plan. Let's go with that. What we can do, Caroline, is with help of other people on this call, is actually help organise that and set a date and do all the logistics around it, rather than than you and your team having to worry about it. We can sort that part of the problem out. We'll keep our fingers crossed for the innovation platform, and we'll go and do our homework about how do we get the Manchester folk across to Shanghai.
I visited Shanghai a few years ago. Absolutely love the city. It's a wonderful place.
It is,
my husband actually is an engineer who worked in Shanghai for a while. So I went there on a holiday and really, really loved it. So yeah, I would love to visit again. Yeah,
no, it is a lovely city. And actually, you know how if you look at the picture behind me, it's a rather grand photograph of the University of Manchester on a sunny day, which is not very realistic, let's be honest. Whereas Catherine's is definitely right. I have heard in that garden, it really does look like that. So yeah, it is a lovely spot. All right. I think rather than prolong the conversation now, I'm conscious that we're close to our closing time anyway. Let us take forward those two actions. One is the half day for Caroline and Ziming's labs to come together and talk about CTC protocols and methods, and how we can actually validate methods across the two sites, and see whether or not you can get very similar results in the two sites. I'm very conscious what you were saying, Caroline, about small cell lung carcinoma being much more prevalent in China than it is in the UK, and therefore clearly an area where we can develop interests very well. The other side of it is Professor Jiao. We should clearly be talking to the colorectal cancer surgery team here in Manchester for you. So we need to take that away and introduce you to Andrew Renahan, Sarah Dwyer, Jorge, and Malcolm Wilson. I again can see opportunities there, but we need to get you with the right people. Okay. All right. I think I'll leave it there and say thank you to everybody for their presentations this morning. It's been really good, really interesting. It's reminded me that I just need to get down the road in Manchester myself. So I will come find you, Caroline.
Well, I'm out in leafy Cheshire, of course, Keith, so it's quite a trek.
Oh, well, okay. Well, I might have to go for that Zoom one then.
Yeah. Okay.
Zoom option, but yes, that'd definitely be worth it. And yeah, thank you ever so much.
Thanks, everyone.
Thank you.